So hello everyone. So today I'm going to show you inoculation of the PUS swab sample and also the type of culture media that you're going to use. If you find this video useful, you can share, you can like, you can comment, and also you can subscribe. Let's go. So let's start by, it is a wound swab, we know. Now the pathogens or the microorganisms that can easily be found in a wound swab. I know you know many of them. I know you know there are many pathogens that can be found in a wound. So I'm going to teach you some of them. Some of them you can go and look for it. So the first pathogen that can be found there is the Staphylococcus species. Mostly Staphylococcus aureus is the common Staphylococcus species that is found in the wound swab, in the wound that is in a wound. So the second type of bacteria, they are called Streptococcus pyogenes. Streptococcus pyogenes. It can be found there. We have an E.coli can also be found there. We have the Proteus species, it can be found there. We have the Klebsiella. We have the enteric bacilli, the enteric bacteria, that is, we have the Acinetobacter, we have the bacteroids, we have the fungi can be found in a wound swab. We have the Mycobacterium, tuberculosis, of course, you know. We have Clostridium, the likes of perfringens. We have the and other types of the Clostridia, but mostly Clostridia perfringens. So those are some of the pathogens that can be found in a wound swab. So after getting that knowledge about the type of pathogens can be found in a wound swab, now is about the selection of a media that can be used for this inoculation of this PUS swab of that sample. So the first type of a media that is very relevant, is commonly used in any type of inoculation, any sample, in any microbiological sample, is the McConkey. The McConkey agar. As you can see here, I have my McConkey agar here, and also I have another one here. This one is the pure McConkey, the plain, and this one is the subital McConkey. You can see the color different. The color different. You can see this one, but mostly both of them are McConkey. Now you can start with the McConkey. I'm going to explain why McConkey. You know the importance of McConkey in this wound swab, the PUS swab, that is. It used to identify those gram-positive bacteria against the gram-negative bacteria. It differentiates between the gram-positive and the gram-negative bacteria. And through this McConkey, you can learn that the gram-positive bacteria, as you know, McConkey is a selective media. It only allows the growth of gram-negative. The gram-positive cannot grow on McConkey. So whenever you inoculate your PUS swab in this McConkey, when you see there is growth, you know actually we are targeting the gram-negative bacteria. The bacteria in that wound swab, it is a gram-negative. And where there is no growth in McConkey, you know that bacteria, it is a gram-positive. So it differentiates. The work of McConkey is to differentiate between gram-positive and the gram-negative. So that is one use of McConkey. The other one use of McConkey is McConkey helps us to differentiate for those lactose fermenters and non-lactose fermenters bacteria. Whenever you learn that your bacteria has produced a type of colony, you can differentiate if it is a lactose or a non-lactose. Of course, you know lactose fermenters, after they utilize the lactose in the McConkey agar, the color changes from red to pink. Those bacteria that are lactose fermenters, they produce a pink color, but non-lactose produce the red color. And for the case of subital McConkey, the colonies, if it is lactose or non-lactose, the lactose color of the bacteria that is there in that sample, the color turns to pink. But if it is non-lactose, the colonies remain colorless. So that is the work of McConkie. So the second type of media that is commonly used, we have the, and very, very important media, we have the chocolate blood agar. This one, as you can see here, this is my chocolate blood agar. And you know, the work of chocolate blood agar, it grows a variety, a wide range of microorganisms. So this the chocolate blood agar gives, if there is growth, shows if there is growth in that sample, if there is any bacteria, if there is any pathogen in that sample or not. Of course, you can identify the growth of that, you can identify if there is any pathogens in that sample, if there is growth in chocolate blood agar. And different microorganisms give different shapes, different sizes, different characteristics on chocolate blood agar. Mostly you can find the most color of the most pathogens that grow in chocolate blood agar, they are cream. They are cream, some are white, some are colorless, but mostly cream is the most color. Then you can check on the size. You can check if the colony is raised or it is flat. So that is the essence of chocolate blood agar, because it allows the variety of different pathogens. Because of course, you know, chocolate blood agar, let me explain a bit. It contains nutrients. The sheep blood agar that is being used to make this chocolate blood agar, it provides nutrients for the growth of bacteria. So any bacteria can grow on the chocolate blood agar. Are we together? So the other type of media that can be used is also the blood agar. We have the chocolate blood agar and we have the blood agar. The blood agar also is very important, because blood agar helps us to differentiate between types of hemolysis. And I had taught about the types of hemolysis in my previous video. If you want to understand more about hemolysis, you can go and check on my other video, the types of hemolysis that occur in the blood agar. So the work of blood agar is to just give us that difference between the type of hemolysis that is being produced by these bacteria. So there are three types of hemolysis, of course. We have the beta hemolysis, we have the gamma hemolysis, and we have the alpha hemolysis. Of course, we know the alpha hemolysis is the one that shows a partial hemolysis. Beta hemolysis show complete hemolysis, while the gamma, there is no hemolysis at all. So when you see your growth in the blood agar, just check that type of hemolysis that has occurred. Yeah, this type of hemolysis, it helps you to identify and helps you to narrow down the type of bacteria that produces that kind of hemolysis, and the type of bacteria, which type of bacteria produces this type of hemolysis. So of course, through that you can understand well, and you know, it can help you to identify that bacteria if you're more curious and more knowledgeable about this. But basically it is just give you types of hemolysis. And also it can be used as a general media because it also has nutrients. So it works the same as chocolate blood agar. The third type of media is the SDA, suboraude dextrose agar. So the suboraude dextrose agar is commonly used for the fungal. If you're suspecting that that wound is affected by the fungal infection, it is mostly used. You can use the SDA to check that type of fungal infection. So SDA is just for fungal infection. And you know, for fungal infection, they are very, very common, because when there is fungal infection, it makes the bacteria come and infest in that type of a wound. So SDA is common, is very important for our case. Then we have the other type of hemolysis, which is the type of media which is nutrient agar, just general. For when you're suspecting Pseudomonas, you can differentiate it from the other by the color of the fluorescence that produced in nutrient agar. So make sure you like, subscribe to my channel. Thank you. Meet you to the next video.